Hi guys, so next topic we are going to discuss is about the sterilization and disinfection. So guys, this topic is very important for your exam point of view and you can expect a number of questions particularly from this topic. So starting with the sterilization. So the sterilization, it is a process by which an article, surface or a medium is free of all the living microorganisms, whether it is in the vegetative form or in the spore form. It means that there is a complete absence of the microorganism. Coming on to the disinfection, the destruction or the removal of all the pathogenic organisms which are capable of giving rise to the infection. This is disinfection, which means that there is a reduction in the microorganism to such a level which is deemed no longer harmful to the health. Fine. So this is the main difference between the sterilization and the disinfection. Now after this, let's discuss the methods of the sterilization as well as the method of the disinfection. Starting with the methods of the sterilization, in this, first we have heat ionizing radiation, in which we have beta electron irradiation and we have gamma photon irradiation. Fine. We have this filtration method. We have the sterilant gases, which includes the ethylene oxide, the formaldehyde. We have the sterilant liquids, which include the glutaraldehyde. Fine. So these are the method of the sterilization. Now discussing the methods of the disinfection. So in this, first we have the heat, the ultraviolet radiation, gases, filtration, chemicals such as alcohols, chlorhexidine, glutaraldehyde, hypochlorites, iodine and phenolic compounds. So now we are going to discuss them one by one, starting with the methods of sterilization. See, we have two methods, the physical methods and the chemical methods. Fine. So here we are going to discuss the physical methods as well as the chemical methods in brief. Starting with the first method, that is the sunlight. So sunlight, it is an active germicidal effect which is due to the content of the ultraviolet rays. Fine. So we are going to take one by one into detail. So second is the heat. So in this we have two types of heat, the dry heat as well as the moist heat. The dry heat, it contains or it consists of four methods: the red heat, flaming, incineration and the hot air oven. And the moist heat, in this we have three methods at a temperature below 100 degrees Celsius. It includes three processes: the pasteurization of the milk, the inspissation and the vaccine bath. Second method is at the temperature of 100 degrees Celsius. As you all know, at the temperature of 100 degrees Celsius, the process is known as the boiling process. Second is the tyndallization and third is the steam sterilizer. Third is at the temperature above 100 degrees Celsius, you can use autoclave. So these are the methods of heat. Third method is the filtration. Fourth one is the radiation, where we have two types of radiations: the non-ionizing radiation, which again consists of infrared rays and the ultraviolet rays, and we have the ionizing radiation, which consists of the gamma rays. So this is all about the physical method. Now coming on to the chemical methods. So the chemicals which we are using for the sterilization or the disinfections are the alcohol, aldehydes, phenols, halogens, salts, surface active antigens, dyes and the gases. So guys, this is a brief about the physical and the chemical method. Now we are going to take one by one in detail. So before going into detail, there are two terminologies. Number one is the antiseptics, What do you mean by antiseptic? These are the chemical agent that slow the growth of the microorganism on the external surfaces of the body, and it helps to prevent the infection. These are antiseptics. So they can be safely applied to the skin or the mucous membrane and are used to prevent the infection by inhibiting the growth of the bacteria. The antiseptics should be distinguished from the antibiotic, important point, as they destroy the microorganism inside the body, and from the disinfectant, which destroy the microorganism found on the non-living objects. Fine. However, the antiseptics are often referred to as skin disinfectant. Commonly asked MCQ. The antiseptics are often referred to as the skin disinfectant. Now, after the antiseptics, let's discuss about the antisepsis. So, it is defined as a prevention of an infection, usually by inhibiting the growth of the bacteria in the wounds or the tissues. So, starting with the physical method, first is the sunlight. So, sunlight, it has an active germicidal effect due to the ultraviolet rays. Fine. So it is a natural method of sterilization in cases of water in the tanks, in the rivers and in the lakes. Fine. It is a natural method of sterilization. Fine. Coming on to the heat, these 2 points are very important. These are the principle of heat. First is the principle of the dry heat. So it kills the organism by denaturation of the bacterial protein, while the principle of the moist heat, it kills the organism by the denaturation and the coagulation of the protein. Important points. This is the main difference between the principle of a dry heat and a moist heat. Now after this, let's discuss the dry heat sterilization. First one is the red heat. So this is the red heat, in which the inoculating wires or the loops, tips of the forceps and needles, these are held in the flame. See, this is the red flame. So these all things, these are held in the flame of a Bunsen burner till they become red hot. Can you see this? These are becoming red hot. So this is the first method of dry heat sterilization, that is the red heat. Now coming on to the second method, which is the flaming. So in this, the glass slides, the scalpels, mouth of the culture tube, these are passed through the Bunsen flame without allowing them to become red hot. Fine. So this is the main difference between the red heat and the flaming, that in the red heat you are held the needles, the inoculating tips, these are held in the flame of a Bunsen burner till they become red hot, while in the flaming you can just pass the glass slides, the mouth of the culture tubes, you are just pass these through the Bunsen flame without allowing them to become red hot. Fine, this is the main difference. Coming on to the third method of the dry heat sterilization, which is the incineration. So it is the effective material. In this, the effective material, it is reduced to ashes by burning. In this, you can put the material or the effective material and it will burnt and it reduces to, or it reduced to ashes. Fine. Examples like soil dressing and beddings. All the infective material is reduced to ashes by burning process. Now last method is the hot air oven. So it is the most widely used method of the dry heat sterilization. The temperature, important, temperature is 160 degrees Celsius for 1 hour. This is the holding time. And in this you can sterilize the glassware products like the glass syringe, pipettes and the test tubes. You can also sterilize the surgical instruments like scalpels, scissors or the forceps. You can also sterilize the chemical products like the liquid paraffin, wax and sulfonamide powder. Important. You just need to remember the temperature, which is 160 degrees Celsius for 1 hour. It is the holding time. Fine. So this is all about the dry heat. As I have already told you, the dry heat, number one is the flaming. First one was the red heat. Red heat, flaming, incineration, hot air oven. In this you can inoculating loop or wires, tip of the forceps and the spatulas. You can sterilize in this. In dry heat you can sterilize the contaminated clothes, the animal carcasses and pathological materials, your swabs, liquid paraffin, dusting powder and fats and grease. And through hot air oven you can sterilize the glassware products, forceps, scissors, scalpels or syringe, all glass syringes, as I've already told you. Fine. So this is all about the dry heat. Now coming on to the second method of sterilization or the physical method of sterilization, which is the moist heat sterilization. As I have already told you that there are three processes: at the temperature below 100, at the temperature of 100 and above 100 degrees Celsius. So in this, first we are going to discuss about the at the temperature below 100 degrees Celsius. So in this, first method is the pasteurization of the milk. So it is a method of sterilization of the milk. It is defined as an heating of every particle of the milk or the milk product to a specific temperature for a specific period of time without allowing the recontamination of that milk or the milk product. So there are two most common methods which are used, and it is commonly asked question, important MCQ point of view. So there are two methods. Number one is the holder method, second is the flash method. The holder method, the time and the temperature is 63 degrees Celsius for 30 minutes, and the flash method 72 degrees Celsius for 15 to 20 seconds. Guys, please note it down, it is important. Now pasteurization kills nearly 90% of the bacteria in the milk, including the more heat resistant tubercle bacillus and the Q fever organism, but it will not kill the thermoduric bacteria nor the bacterial spores. Important. Fine. So this was the first method of the moist heat sterilization at the temperature below 100 degrees Celsius. So the second method is the inspissation. So in this, the serum or the egg media, which is the LJ, which means the Lowenstein-Jensen's and the Loeffler serum, are sterile by heating at 80 to 85 degrees Celsius for half an hour on the 3 consecutive days. Important. The time and temperature. See, the time and temperature in the sterilization and disinfection is very important. Fine. Now third one and the last one is the vaccine bath, in which the bacterial vaccines, these are sterilized at 60 degrees Celsius for 1 hour. Important. Vaccine bath is just for the sterilization of the bacterial vaccines at the temperature of 60 degrees Celsius for 1 hour. Now the next method is at the temperature of 100 degrees Celsius. First method is the boiling. As you know, it usually occurs at 100 degrees Celsius. So boiling for 10 to 30 minutes may kill the vegetative form, but many spores withstands. Important. Second in this is sterilization. So in this, the steam at 100 degrees Celsius for 20 minutes on 3 consecutive days used for the sterilization. This is the sterilization. Sterilization of the egg, serum, sugar containing media. Important. And last is the steam sterilizer at 100 degrees Celsius for 90 minutes. It is also known as Koch's or the Arnold's steam sterilizer. Fine. These all points are very important for your MCQ point of view. Now last is at the temperature above 100 degrees Celsius. In this you can use the autoclave. So the autoclaving is a method to sterilize the equipment by subjecting them to high pressure, which is 121 degrees Celsius. Very important. So this is a sterilization by the steam under pressure. Guys, please note it down. It is steam under pressure, SUP, steam under pressure. The device which is used for autoclaving is known as autoclave. Autoclaving is used for the culture media, the dressing and the aprons, the surgical instrument except the sharp instruments, gloves and the catheters. Autoclaving steam under pressure can kill the bacterial spore. Important. Nothing short of autoclaving at high pressure can destroy the spores and ensure the sterilization. This table is very important. This is the distinguishing feature between the autoclave and a temperature and the time of an hot air oven and autoclave. So autoclave, it occurs at 3 temperatures. It works on 3 temperatures. Number 1 is, which is very common, 121 degrees Celsius and with the holding time of 15 minutes, 126 degrees Celsius with a holding time of 10 minutes, 134 degrees Celsius with a holding time of 3 minutes. As much as you can increase the temperature, the time decreases. And in the hot air oven, 160 degrees Celsius leads to 45 minutes, 170 degrees Celsius leads to 18 minutes, 180 degrees Celsius leads to 7.5 minutes and 190 degrees Celsius leads to 1.5 minutes. Important. Actually very important. Fine. Now this is all about the moist heat. As I've already told you, the temperature below 100 degrees Celsius, pasteurization of milk, 2 methods, Holder method, flash method, and inspissator, in which the heating of Lg, Lowenstein Jensen medium, and the Loeffler serum for 3 consecutive days or the successive days. Moist heat, second is the temperature at 100 degrees Celsius, as you know, boiling culture media. Steam at atmospheric, sterilization of the dressing apron, sterilization. Steam under pressure, which is the autoclaving. Most of the culture media, gloves, surgical instrument except the sharp instrument, suture material except the catgut. So this is all about the dry heat and the moist heat. Now third method of sterilization on the physical method is the radiations. So in this we have 2 types of radiation, the ionizing radiation and the non-ionizing radiation. See, in the non-ionizing radiation we have the infrared radiation and the ultraviolet radiation. Infrared radiation, it is the pre-packed items such as the syringes and catheter. It sterilizes the pre-packed item, while the ultraviolet radiation, it enters the way, operation theaters and laboratories. Fine. Second is the ionizing radiation or the gamma radiation. It is an heat labile instrument. It is used to sterilize the heat labile instrument like the plastic, syringes, swabs, greases, surgical catgut, bone and the tissue graft. Important point is the sterilization by the ionizing radiation is called as cold sterilization. Important. What is cold sterilization? The sterilization by the ionizing radiation is known as cold sterilization. So last method in the physical methods are the filtration. So in this, it is used to sterilize the syrup, the sugar and the antibiotic solutions. Types of filters are: number 1, the earthenware candles. And in this we have two types of candle filters: the diatomaceous earth filters, example is the Berkfield, and the unglazed porcelain filters, example is Chamberland. Example is Chamberland, important. Second type of filter is the asbestos disc filters, also known as Seeds filter, important. It is made up of the magnesium silicate. Third are the sintered glass filters. Fourth one are the membrane filters. These are made up of cellulose esters, nitrocellulose or the millipore, important, which is widely used, and it is with the size of 0.015 to 12 micrometer, important. Fine. And this is commonly asked question, this is commonly asked question, and this is commonly asked question. So this was all about the physical method. Now coming on to the chemical methods. So in this, first is the alcohol. So in this we can use the ethyl alcohol, isopropyl alcohol. These are act by the denaturation of bacterial protein. These are used as a concentration of 60 to 70% in the water. The important point, the methyl alcohol, it is effective against the fungal spores. Important for your MCQ. Second are the aldehydes, in which we have the formaldehyde and the glutaraldehyde. So in the formaldehyde, it is bactericidal, sporicidal and the virucidal. 10% of aqueous solution of formalin is used to sterilize. Fine. Coming on to the second aldehyde, that is the glutaraldehyde. It is an effective against the Mycobacterium tuberculosis, the fungi and the viruses. It is used as 2% of the buffer solution as the Sidex, important, commonly asked. So sterilization of it is mainly used for the sterilization of cytoscopes, the endoscopes and the bronchoscopes. Next are the phenols. In this we have the cresol, we have the chlorhexidine, we have the chloroxylenol. Fine. Starting with the cresol. So Lysol, it is a solution of the cresol in the soaps. It is used to sterilize the infected glassware, cleaning floors. While the chlorhexidine, it is a Savlon chlorhexidine plus citramide. It is used as disinfection of skin, important. Bactericidal at high dilution. Next is the chloroxylenol. It is an active ingredient of Dettol. Next are the halogens. So in this, the chlorine and the iodine, which are used as an disinfectant. Next are the salts. The salts of the copper, silver and mercury, these are used as an disinfectants. Coming on to the surface active agents. So in this we have 4 types of surface active agents: the anionic, cationic, non-ionic and amphoteric compounds. So in anionic, it is commonly used as a soap and detergents. Cationic, quaternary ammonium compounds which are effective in rabies virus, important. In amphoteric compounds, it is also known as Tego compounds. Tego compounds. Coming on to the dyes. So in this we have 2 types of dyes, the aniline dye and the acridine dye. So the aniline dye, it includes the crystal violet, the brilliant green and the malachite green. Fine. The crystal violet, the brilliant green and the malachite green. These are more effective, or these are more active against the gram-positive bacteria. Fine. While the acridine dyes, these includes in the acriflavin and the proflavin, and these are also more active against the gram-positive bacteria than that of gram-negative bacteria. Last are the gases. So in this we have three types of gases: the formaldehyde, the ethylene oxide and the BPL. First is the formaldehyde gas. So the formaldehyde gas, it is generated by adding the KMnO4 to formalin. Important. The sterilization is done by condensation of the gas on the exposed surface. This is the method of sterilization. You have to condense the gas on the exposed surface, the surface which has to be sterilized. Next is the ethylene oxide. These all points are very important. Ethylene oxide, highly penetrating gas. It is highly inflammable, causes mutagenicity and causes the carcinogenicity. Important points. Last is the BPL, which means the beta propiolactone. It is used as 0.2% and it is used for inactivation of vaccines. Fine. So this is all about the sterilization and the disinfection. Now after this, let's discuss some important points, or the points to remember, which are very important for your exam point of view, because these are itself act as an MCQs. Coming on to the procedure of sterilization of some important material. So in this, first is the inoculating. As you all know, inoculating loop, wire and tip of the forceps and the sealing spatulas. Method of sterilization is flaming. Contaminated cloth, pathological material, incineration. Glassware, syringes, petri dishes, test tubes, flask, surgical instrument, hot air oven. Most of the culture medias, these are sterilized by autoclaving or by membrane filter. The sharp instruments, these are sterilized by 5% of cresol or the hot air oven. The clinical thermometers, these are sterilized by Savlon. The cytoscopes, bronchoscopes and other endoscope, corrugated rubber, anesthetic tubes, face mask, plastic endotracheal tubes, these are sterilized with the help of glutaraldehyde. Very important MCQ. Fine. Milk gets sterilized with the method of pasteurization. The suture material except the catgut, autoclaving. Catgut by ionizing radiation. Bacterial vaccine by vaccine bath. We have already discussed all these points. The dressing, aprons, gloves, catheters, surgical instrument except the sharp instrument by autoclaving. Heat sensitive catheters, wards, fumigation, laboratories, sick room operation theaters by formaldehyde gas and ultraviolet rays. The pre-packed items such as syringes and catheter by infrared radiation. The culture media containing the gelatin and sugar by tenderization. Next important points are all the suture material except the catgut, autoclave, rubber, gloves, surgical instrument and most of the culture media by autoclave, catgut by ionizing radiation, forceps, scalpel, scissors and needles by hot air oven, disposable syringes by ethylene oxide. Important. Suture materials by glutaraldehyde, hospital waste by incinerator, which is burning and leaves into ashes, vaccines, serum, antibiotic solution by filtration, operation theaters by fumigation with formaldehyde gas, preparation of the skin for the surgery by iodine, spirit, Savlon and chlorhexidine, absorbent point Broaches and files, important, by hot salt sterilizer or the glass bead sterilizer. Guys, very important. Cotton pliers, cement spatulas, by passing the working blade quickly through the flame several times. The depend dish and the glass slabs, swabbing the surface of the tincture of thermoseal followed by double swabbing with alcohol. GP con, very important, commonly asked MCQ. GP con, by immersing 5.2% of sodium hypochlorite for 1 minute followed by rinsing with the H2O2 sodium peroxide and drying. The silver cones, slowly passing them back and forth through the Bunsen burner for 3 to 4 times by immersion in the hot salt sterilizer for 5 seconds. Loeffler serum slope or the Lowenstein-Jensen medium through inspissator. Sugar media through tyndallization or intermittent sterilization. GMT, enamel hedges, corrosive sensitive burrs, orthodontic pliers by Autoclave. Cheetle forceps by 1% of Savlon. Knife and scissors soaking in Lysol. ZOE polyether impression material with the help of 1% of glutaldehyde. Alginate polysulfide condensation and addition silicone impression material 1% sodium hypochlorite. Acrylic denture can sterilize in iodoform. Suction tubes, endoscopes, bronchoscope, these are sterilized in Sidex, which is 2% of glutaldehyde. Culture media by fraction sterilization. Pre-packed material by X-rays. Plastic surgical needle and sutures by gamma rays. Pumice by sodium hypochlorite. Very important table. This covers all your sterilization, method of sterilization, disinfection. Last is, which is commonly asked, the biological indicators. Guys, the biological indicators, these are the standardized preparations of the microorganism which is used to assess the effectiveness of a sterilization process. They usually consist of a population of bacterial spores which are placed on an inert carrier, for example, a strip of a filter paper, a glass slide, or a plastic tube. Most commonly, spores of Bacillus stereothermophilus or the Geobacillus stereothermophilus are used. Very important. Most commonly, spores of Bacillus thermophilus, Geobacillus stereothermophilus, these are used. Spores of Bacillus subtilis, Bacillus pumilis are also used. This line is very important. So, for various methods of sterilization, following biological indicators are used. The moist heat autoclave, spore used are Bacillus stereothermophilus. Dry heat hot air oven, Bacillus subtilis. Ionizing radiation, Bacillus pumilis. Gas sterilization with the help of ethylene oxide, Bacillus subtilis. Plasma sterilization, Bacillus stereothermophilus. Plasma sterilization is important, Bacillus stereothermophilus. Fine. So guys, this is very important topic, and this is all about the sterilization and disinfection. Please go through it once. Thank you.